So my lab has been interested in coronavirus entry into cells for a number of years, and with the focus on spike-mediated conformational changes. And I will be telling you some of the work we've been doing since the emergence of SARS-CoV-2 and how that led us to design a vaccine using an approach that's quite different from what big pharmas have been following. So as we all know, SARS-CoV-2 emerged at the end of 2019, and after a few weeks, my lab was able to develop one of the first pseudovirus entry assay for this virus. That work was done by Lexi Walls in the lab, who was able to show within 2 weeks of the release of the sequence that ACE2 rendered cells susceptible to cell entry. And she worked very closely with Youngjun Park in my lab to determine structures of the spike glycoprotein using Cryo-EM. And we observed multiple conformational states from a single data set. What Young saw is that the receptor binding domains, or RBDs, that are the apex of the spike, could adopt closed and open conformations, which are similar conformational changes to what we had observed previously for SARS-CoV and MERS-Cov. And opening of one receptor binding domain allows engagement of this domain with the ACE2 receptor. So these structures really showed that the entry mechanism was probably quite similar in terms of conformational changes between SARS-CoV-2 and SARS-CoV-1 and MERS-Cov, and they've really been used as a blueprints by thousands of groups worldwide to map the emerging of mutants, as well as to develop vaccines and therapeutics. So one of the first questions we were trying to answer was, what is the nature and the specificity of antibody responses in infected individuals? And this is work we did in collaboration with David Ecorti at Vir Biotech, and we delineated an antigenic map of the SARS-CoV-2 receptor binding domain that I will be calling RBD, which is illustrated here on the left-hand side with antibody Fab fragments shown in different colors. So we solved a number of structures showing that multiple different epitopes could be recognized, and we classified them in antigenic sites based on competition binding. And using serology in parallel to isolation of monoclonal antibodies, we could quantify the magnitude of antibody responses at each site. For example, site 1, shown with 2 antibodies in green and orange, is the ACE2 receptor binding site and is immunodominant. Most antibodies in the plasma of infected individuals are actually targeting this site, which outcompete the receptor, and therefore there's a correlation between antibodies blocking ACE2 binding and neutralization in vitro. So to understand the quantitative contribution of RBD-specific antibodies to neutralization, we asked what will happen if we deplete all the RBD targeting antibodies from the plasma of infected individuals. And we did exactly that and measured neutralization before and after depletion. And what we can see that we have significant drop, about 10-fold reduction in serum neutralizing activity upon doing that, showing that the RBD is not only the target of many antibodies in the plasma or memory B cells of infected individuals, but it is quantitatively the most important target of neutralizing antibodies. In the interest of time, I will highlight just a couple of antibodies that are of special interest, starting with the one shown on purple, which is called S309 or Sotrovimab now. And this antibody was actually isolated at the very beginning of the pandemic from a SARS-CoV-1 infected individual back in 2003, from whom we had already characterized an antibody specific for SARS-CoV-1. In this case, from these samples that our collaborator, David Ecorti, had, we were trying to find cross-reactivity and neutralization between SARS-CoV-1 and SARS-CoV-2. And the very large screening allowed to find S309, which we showed blocks spike-mediated entry of SARS-CoV-2 and multiple isolates of SARS-CoV-1, as well as the Bat virus related to SARS-CoV-1, named WIV1. Using a SARS-CoV-2 challenge model in hamsters, we were able to show that administration of S309 in a prophylactic setting 1 day before infection was protective even at very low dose. We have complete shutdown of viral replication in the lungs of hamsters at 1 mg, at doses as low as 1 mg per kg. So using Cryo-EM, we determined the structure of the complex with the spike and found that the antibody recognizes the receptor binding domain to an epitope that is accessible in both the open and closed states. And therefore, the antibody does not discriminate between the different conformational states of the RBD. And if we zoom in at the interface, there is a highly conserved glycan at position N343, which is actually integral part of the epitope. And as you can see on the right-hand side, the core fucose is sandwiched at the interface between the heavy and light chains, and about 25% of the surface buried upon binding of the antibody is actually accounted for by the glycan. This glycan is highly conserved, and its removal is actually negatively affecting folding. So if we look at the sequence conservation across SARS-CoV viruses, we see that the epitope recognized by S309 is highly conserved, as shown by this dark blue color, and only a couple of residues at the periphery of the epitope are conservatively substituted. So because this antibody targets such a highly conserved epitope, which is conserved across different SARS-CoV viruses, it is actually completely resilient to the emergence of SARS-CoV-2 variants. This epitope is not changing at all. And a few months ago, we had the very positive outcome of the clinical trial that the antibody entered, showing that it reduced hospitalization and death by 85% compared to placebo. And this antibody received an emergency use authorization from the FDA back in May, and it's used in the clinic worldwide. It has already been approved in Australia, for example. So that was really, really exciting results for us, and I think it defines a paradigm shift of how to look for antibodies, as opposed to look in an individual or individuals who have been infected by the pathogen of interest. Here, by starting with a SARS-CoV-1 infected individual, we ended up finding an antibody targeting a highly conserved site, as opposed to some of the epitopes that are very sensitive to variants. So S309 neutralizes viruses belonging to clade 1A and 1B that cover SARS-CoV-1 and SARS-CoV-2, but it does not cross-react. It does not cross-react with viruses belonging to the other two clades of the SARS-CoV virus subgenus. A legitimate question is, how much should we be worried about these viruses, the ones that are not similar to SARS-CoV-2 and SARS-CoV-1? So Sam, a grad student in my lab, decided to answer this question, and she teamed up with Tyler Starr in Jesse Bloom's lab to try to understand if viruses from other clades could actually use ACE2 and could potentially use human ACE2, which would suggest that they are poised for human infection. And I will be talking about the second virus. I don't think you see my pointer, but the second virus from the pink clade, Bat-72, in which Tyler and Sam were able to identify that it can bind Rhinolophus affinis ACE2, especially one of the two alleles, the one that is at 9479. And in the pseudovirus entry assay, we could see that expression of this Rhinolophus affinis ACE2 allowed efficient spike-mediated entry into cells. Tyler and Sam identified that introduction of 2 amino acid mutations in the RBD of the Bat-72 spike even allowed usage of human ACE2 for spike-mediated entry, suggesting that it does not take much for these viruses to actually be able to use human ACE2. And perhaps we should find countermeasures that are efficient against these more divergent clades as well. So we did exactly that. Ali in the lab found an antibody that cross-reacts with all 4 clades of the SARS-CoV virus subgenus called S2X259, and determine the structure of this antibody bound to the spike. And here, focusing on just the RBD part, we can see that the antibody binds onto the core RBD, away from the receptor binding motif, mostly away, to an epitope that is highly conserved across SARS-CoV viruses, explaining cross-reactivity. And it remains away from all the mutational hotspots shown in red that are found in SARS-CoV-2 variants. So the antibody actually neutralizes all SARS-CoV viruses we have an assay for, including Bat-72, and is therefore a lot broader than S309, even broader than 309, and is a good clinical candidate, not only for the current pandemic, but for pandemic preparedness should a new SARS-CoV virus emerges into humans. So if we map the binding site of S2X259 on the RBD as compared to ACE2, we can see that they actually share a few residues in common around position 501, and therefore the antibody blocks ACE2 attachment competitively as a mechanism of inhibition. We showed that S2X259 was protective in vivo in a therapeutic setting at 5 mg/kg with complete inhibition of viral replication in the lungs, and we have partial inhibition at 1 mg/kg. And the same holds true in a therapeutic setting by giving the antibody a day after viral exposure, and at similar doses with very efficient protection. So far I told you only about the RBD, but we have been interested in understanding what's going on with the NTD. And Matt, a very talented postdoc in the lab, realized early on that among the antibodies cloned from memory B cells of infected individuals, although most are targeting the RBD, a significant fraction, 5 to 20%, were actually accounted for by NTD targeting antibodies. Matt delineated an antigenic map of the SARS-CoV-2 NTD that is shown here with 5 fragments in different colors, and observed that only antibodies targeting the site shown in red were actually neutralizing, and we call that an antigenic supersite. And we show that this type of antibodies are also protective in vivo in the hamster challenge model. At 1 mg/kg or 4 mg/kg, we have inhibition of viral replication, except for a few animals, as you can see, for which there is no difference with the control group. And we wondered, what's going on here? And we decided to sequence the virus from the lungs of these animals. And what we observed was they all shared in common mutations comprising a deletion of residue Y144. This maps exactly at the center of the epitope recognized by this type of antibodies, and it was actually present in the alpha variant. So that showed that we recapitulated in the hamster model the clinical viral evolution that is ongoing in clinical samples. And indeed, when we delete residue 144, we have complete abrogation of binding and neutralization by these types of antibodies. So escape mutants had arisen in this model, whereas they were not part of the challenge stock. So we have structurally characterized a fair number of these variants. And here I'm showing you NTD structures. And actually, I'd like to draw your attention on the most bottom row, showing a superimposition and then a single structure at a time of the wild type, kappa, delta, and epsilon variants, where the region comprising 144 is actually quite different across these structures, because the NTD is very plastic, mutating and changing its structural organization, which seems to be very efficient to evade antibody neutralization without any effect we can really measure or detect on the function of the spike. And here I'm showing you a heat map of ELISA binding into the different variants from a panel of neutralizing antibodies binding the NTD. And you see that most of them have reduced binding by multiple orders of magnitude to all of these variants. So we think the NTD is important, and it is targeted by neutralizing antibodies. That's why it's actually mutating. But it seems to be a very challenging target to focus on due to its ability to evolve so rapidly. So based on our work, we thought that it might make sense to try to focus antibody responses on the RBD, which is clearly the main target of neutralizing antibodies in the plasma and memory B cells of infected and vaccinated individuals. The problem is that the RBD is just a small domain and it's not very immunogenic. So to try to solve that problem, like in our lab, teamed up with Neil King here in our department at the UW, to display the RBD as a genetic fusion to a trimeric protein, which can be mixed with a pentameric protein. Both of these trimeric and pentameric building blocks were designed in Neil King's lab to self-assemble into an icosahedral nanoparticle displaying 60 copies of the RBD in a flexible manner. In a preclinical study in mice, we showed that various flavors of these nanoparticles were eliciting about 10 times higher titers of neutralizing antibodies compared to the prefusion stabilized spike, and which you can see on the y-axis, the higher, the more potent neutralization. And in a large non-human primate study, in collaboration with Vali Pulendran at Stanford, we showed that our nanoparticles formulated with multiple clinical adjuvants were protective against SARS-CoV-2 challenge, against viral replication in the upper and lower respiratory tract, as well as against signs of disease in the lungs, as shown on these PET scans, as opposed to the no vaccine control. So with very strong support from the Gates Foundation and CEPI, the vaccine, which is the name GBP510, has entered phase 3 clinical trials a few months ago. And we are actually quite excited about the possibility that it could make a difference due to its high shelf life stability and the ability to manufacture at scale. So a question that I'm oftentimes asked is how good are current vaccines against divergent SARS-CoV viruses? And I think one of the best answers to that question comes from a paper published by Ralph Berrick's lab, in which they designed multiple flavors of chimeric spikes, but the only data I would like to emphasize are the ones shown in red, group 4, in which we can see that in vaccination of mice with the spike mRNA vaccine that encodes for the SARS-CoV-2 spike elicits high titers of neutralizing antibodies against SARS-CoV-2 that are reduced by about 1.5 orders of magnitude against SARS-CoV, and there is no detectable neutralization against RS-SHCO14. So suggesting that current vaccines might not be great, and similar results have been found by we and others using various platforms. So breadth against divergent SARS-CoV viruses is probably not something we should be expecting from our current vaccines. And actually, upon challenge of these mice with SARS-CoV-1, Ralph's group observed that, as shown in red, the protection afforded by SARS-CoV-2 spike vaccination is almost equivalent to vaccination with a control group with a norovirus capsid. So there is really no protection against a divergent SARS-CoV virus. So one of the things to keep in mind is that all the broadly neutralizing SARS-CoV virus antibodies found to date, these are the ones we worked on, target the RBD, suggesting the RBD is probably still a good target for eliciting broad immunity against SARS-CoV viruses. And actually, we very recently found an antibody that was not on the previous slide that recognizes the receptor binding motif through contacting residues that are exactly the same. Almost all residues, about 3/4 of them, are the same residues as the ones contacted by ACE2. So this antibody is mimicking the ACE2 footprints on the RBM, which allows it to cross-react and broadly neutralize not only SARS-CoV-2-like viruses, but SARS-CoV-1 and related viruses. And it's very interesting because if you look by deep mutational scanning to the potential residues that can be introduced to a Brogade binding, you can see that there are few of them, and all of them are actually associated with a severe dampening of ACE2 binding. In collaboration with Sean Whelan, we did viral passaging to isolate escape mutants, and only the H at position 489, shown here at the bottom right, popped out. But as you can see, based on the color scale, there is an almost 10-fold reduction in ACE2 binding. And when we do a viral competition assay between the wild-type virus and this escape mutant 489H, after 4 cycles, the escape mutant is completely outcompeted because it's not fit enough. So it's almost impossible to escape neutralization from this antibody due to the functional constraints by contacting the ACE2 binding residues specifically. So based on that knowledge of targeting of the RBD by multiple SARS-CoV virus bNAbs, we went back to the drawing board and decided to design a vaccine that will potentially elicit these types of antibodies specifically. And to do that, we thought that we should present multiple different SARS-CoV virus RBDs on the same nanoparticle to form a mosaic that will present different RBDs next to each other, with the goal of eliciting antibodies that are cross-reactive across these antibodies. As I briefly mentioned a few slides ago, these nanoparticles are stable at room temperature. Here we see in the 3 rows, 3 different metrics to follow that absorbance, dynamic light scattering and retention of ACE2 binding, and only one of the 4 formulations is actually showing a little bit of aggregation at 37 degrees. But even at 28 degrees, 25 degrees, the particles are stable on the bed for over a month. So vaccination of mice with this mosaic, we have various flavors of mosaic containing 3 or 4 RBDs, and just in the interest of time, I just consider them all together to show that they elicit broadly neutralizing antibodies, neutralizing SARS-CoV-2 on the left-hand side. And the 4 groups shown, I apologize, my pointer doesn't show up, but the groups shown in purple and pink are equivalent to the homotypic neutralization shown by the light blue group for SARS-CoV-2 on the left, and compared to the green group for SARS-CoV-1 on the right-hand side. So in collaboration with Ralph Baric, we challenged these mice with SARS-CoV-1, and we saw complete protection from viral replication in the lung using the mosaic with 4 RBDs. And that remained true even when we removed the SARS-CoV-1 RBD from the mosaic, providing proof of principle that these mosaic RBD nanoparticles can actually elicit broad sarbecovirus immunity, protecting against heterotypic challenge. How am I doing time-wise?
A couple of minutes, we're good. Yeah.
Okay. That's what I like to think about version 2.0 of our vaccine, and we're actually working on optimizing the composition of the RBDs to move it to the clinic. But oftentimes people ask me about universal coronavirus vaccine, and I think this is coming from the flu field. And so I felt like maybe I should share use the last few minutes to share the state of the field and where I think we're headed. So Max Sauer in the lab identified an antibody a few years ago now that recognizes the stem helix, which is an epitope found at the proximal parts of the spike near the fusion machinery. And this antibody was elicited by vaccinating mice with the SARS-CoV and the MERS-Cov spike glycoprotein. We determined, in addition to a cryo-EM structure, multiple crystal structures of this antibody bound to the peptide epitope it recognizes from SARS-CoV, from MERS-Cov, and OC43. And we realized that this epitope is highly conserved among beta coronaviruses, explaining the cross-reactivity and the ability of this antibody to neutralize MERS-Cov and M-Cov, but it did not bind strongly enough to SARS-Cov to neutralize them. But we subsequently found a similar antibody from an infected individual in collaboration with David Corti. This antibody it has greater neutralization properties because it can actually neutralize SARS-CoV, MERS-Cov, and M-Cov, although with weaker potency. About the IC50 is around 1 microgram per ml, whereas best-in-class RBD-specific antibodies are around 1 nanogram per ml. In the same hamster challenge model, we looked at viral loads upon challenge using either the antibody in the, with a human sequence, a human backbone labeled HU, or with a hamster backbone, which will allow engagement of hamster Fc gamma receptor. And what we saw is that the hamster version provided an order of magnitude reduction in viral load compared to the human version, suggesting that although these antibodies are able to reduce partially viral load, they are not as efficient as RBD-specific antibodies, but it appears that effector function play a key role in reducing viral load for this type of antibodies. And that correlates with the fact that we were able to measure antibody-dependent cellular cytotoxicity and phagocytosis in vitro. One of the limitations with these antibodies is that when we look at their frequency in the memory B cell of infected individuals, we see that they are very rare. Antibodies reacting with the stem helix peptide of one of the 4 viruses shown are present at less than 1% in convalescent individuals, and at most a couple % in vaccinated individuals. So the challenge is that we are looking at antibodies that are much less potent and a lot more rare than RBD-specific antibodies, but with the desired property of neutralizing multiple highly divergent coronaviruses. And in the last slide, I would like to share some data that we recently obtained in collaboration with Federica Salusto, and the work was led by Alejandra in my lab. And we found antibodies that were here able to neutralize not only beta coronaviruses, but alpha coronaviruses, including NN63 and 229E. And we determined the crystal structure of the Fab fragment in complex with the peptide targets, which turns out to be the fusion peptide, which is really highly conserved, and that we had predicted 6 years ago could actually be a very good target for neutralization. If we superimpose the crystal structure shown as a surface representation with the spike trimer, we see that they are not compatible. The antibody completely overlaps with the spike trimer. And we know this region has to be dynamic, and there's probably molecular breathing going on that will allow the protease TMPRSS2 to activate the spike just before membrane fusion takes place. And that's actually a mystery how these antibodies are elicited. Is it due to transient exposure of that site at one point during the viral entry cycle? That's most likely, but we are working on that to try to figure it out. Just wanted to share with you these latest results, and just to emphasize that these antibodies have a potency that is similar to stem helix antibodies and a frequency that is as low, if not lower. So again, although this is paving the way towards designing broad coronavirus vaccines, the challenge ahead of us will be to figure out how to elicit high titers of these antibodies to compensate for the fact that they are much weaker neutralizers than best-in-class RBD antibodies. So I would like to thank the members of my lab who did the work I mentioned throughout the pandemic, and I showcased them during on each slide, and our wonderful collaborators, which I also mentioned throughout the presentation. Thank you for your attention. I'll be happy to take any questions. And again, I apologize for the glitch at the very beginning.
David, thank you. Beautiful, beautiful talk. Yeah, your talk, highlights the ability to have dynamite monoclonals that do exactly what you would love, and of course to use those therapeutically. But then the trick is how to elicit them at high affinity and high prevalence is the challenge, and that will be the trick to try to figure out how to make that happen.
Yes, for broad beta coronavirus neutralization, or even broader, definitely. I think for broad Sarbecovirus immunity, I think we're almost there, and I really think that our approach and the one that Ralph Berrick's lab shows, which is completely different but also very, very effective. I think we have multiple approaches that are poised to working to elicit broad Sarbecovirus immunity. I'm very reluctant to use the word pan-Sarbecovirus because we know that we have probably sampled a small fraction of the diversity of Sarbecoviruses, so we are not even aware of the full extent of the diversity. But broad Sarbecovirus immunity, I'm pretty comfortable with that term, and thinking that we are poised to making this happen. For broad beta coronavirus or universal coronavirus vaccine, I think we have a long way to go.
Well, the nanoparticles seem beautiful, and the mosaic idea is very intriguing. That that you can elicit, you can focus things to breadth, maybe in that way.
Yeah, we hope so.
So we have one question in the box from John Ming P. Wonderful talk. I'm wondering why you choose the 2-component 15350 nanoparticle over other single component nanoparticles, such as ferritin and I301. I don't know those, of course. So, I mean, there are a few different reasons. One of them is that we have had success with that nanoparticle in the past for other systems. We work with Neil King, who developed it, and it worked very well for RSV. There's actually a candidate RSV vaccine that has entered phase 1 clinical trial using the same nanoparticle platform. And I think the genesis of this project was really our emerging understanding of antibody responses, and I wanted an RBD vaccine, and the platform that Neil King's lab had developed allowed us to really jump into that at the very beginning. So that was the main determining factor.
Yeah.
I actually have a question for you, David. Could you comment on where you are in terms of establishing clinical trials for the RBD nanoparticles?
So I
Is
can
that
share
something in our near future, or
yes.
or is it further off?
So the RBD nanoparticle is already in phase 3 clinical trials. We are working with a company in South Korea called SK, and the project is funded by CEPI. So they completed phase 1-2 clinical trials about mid 2021, and phase 3 is already pretty far along. So we are I can't predict when we will get the results from phase 3, but I think it's not unreasonable to expect that it will come by the end of the year.
Great, thank you.
No, David, I had one question, which is, in all these nanoparticles with other proteins involved, does one worry about response to those proteins?
We think about it, and we actually see a response against the nanoparticle itself. And we have even seen a correlation between response against the nanoparticle and neutralizing antibody titers. So it does not seem to be a problem. And this is something we had looked at with RSV as well a few years ago, but we definitely keep track of that and wonder how it could affect the reuse of that platform for multiple completely different vaccines.
Yeah.
It may or may not affect it. So far, we clearly see antibody responses that are pretty strong against the platform itself, but that do not seem to be a problem at all.